Welcome to BioBits and Bytes. Today we're zooming in on a revolution that's fundamentally changing how we see life itself, promising to unlock secrets about human health that have been, well, hidden in plain sight.
It really is more than just a shift in perspective. I'd call it an evolution in spatial awareness. For decades, biology has been operating with, one hand tied behind its back.
Right.
And now we're finally seeing the full picture.
Absolutely. Think about your body or, any complex biological system as this large, bustling, complex metropolis. For years, our best scientists have been trying to understand how this city works, using methods that are essentially shortcuts.
And we've had incredible success with those shortcuts, don't get me wrong. Bulk sequencing gave us the complete library of all the cell types and molecules present.
Like knowing the phone book and the inventory of every store in the city.
Exactly. And single-cell sequencing allowed us to isolate individual residents and figure out their job descriptions.
But, and this is the key part, if you take all the people and buildings out of the city, put them in a giant blender, and then analyze the resulting mash, you lose the critical context.
You lose everything.
You lose the flow of traffic, the physical proximity of, say, the central bank to the stock exchange, the hidden conversations in a specific alleyway. You lose the neighborhood.
And that jump from a flat, two-dimensional inventory list to an immersive, functional, three-dimensional reality, that is the core mission of modern biology. We need to see the organization, the structure, the communication networks as they exist in situ.
That brings us to our mission for this deep dive. We're unpacking whole-body spatial omics, or WBSO, and the groundbreaking technologies that allow scientists to map molecules, DNA, RNA, protein in full 3D context.
And not just in a thin slice of tissue.
No, within intact tissues, organs, and eventually whole organisms. This isn't just spatial transcriptomics. This is really taking it to the macro scale.
The foundational problem we're fixing here is the loss of the where. Biological inquiry, at its heart, involves 4 dimensions: who, what, when, and where.
Who, what, when, where.
Right. Traditional destructive methods, they sacrifice the spatial context. When you destroy tissue structure to count molecules, you get fantastic resolution on the who and the what, but you lose the intricate details of cell communication.
You lose the structural mechanics of tissue maintenance and, I guess, critically, how diseases physically spread or are contained.
Precisely. So losing the spatial context means we might understand the parts, but we completely miss the functional architecture.
It's the difference between knowing all the components of the skyscraper and knowing why that skyscraper is structurally sound and how it interacts with the buildings around it.
That's a great analogy. For example, in oncology, we might detect that a powerful immune cell is present in a tumor, but if we don't know that it's physically trapped by dense stroma and unable to penetrate the tumor core,
then we miss the functional failure point.
We miss the entire story. WBSO is designed to bring that crucial where back to the forefront of discovery.
Okay, let's unpack this. Our material organizes this whole revolution into 3 necessary breakthroughs.
First, we have to look smaller than the cell itself. We need to understand the fundamental architecture of the 3D genome, the folding of the nucleus's blueprint, and how that structure actually controls gene function.
So you can't map the house if you don't understand the wiring diagram.
Exactly. Second, we'll dive into the next generation of high-resolution, high-throughput spatial technologies that are emerging to capture this data. This means distinguishing between existing optical technologies, which are, limited,
and these revolutionary non-optical techniques, which are enabling true whole-body scale.
Correct. And finally, we will tackle the massive data challenge that arises when you are no longer generating megabytes, but terabytes of high-dimensional information in a single scan.
And that's where computing, AI, and specialized algorithms have to step in to tame this big data problem and ultimately translate these maps into new drug targets.
It's an integration challenge of physics, chemistry, biology, and computer science all rolled into one.
Have you ever wondered why analyzing cells one by one misses the bigger picture? That loss of the cellular neighborhood, that social network, is exactly what WBSO is fixing now. And to start our dive, we have to look at the blueprint right inside the nucleus to really understand the importance of 3 dimensions.
Let's do it.
So we start inside the cell nucleus. When we think about genetics, most people think about the linear sequence. A, T, C, G, the flat line of text that encodes life.
The standard model.
Right, that's what linear sequencing gives us. But our sources really emphasize that the genome is not a flat line. It's a massive, complex piece of folded molecular origami.
That origami analogy is absolutely essential. The 3D genome organization is the study of how that immense linear DNA, which, if you stretched it out, is about 2 meters long.
2 meters.
2 meters long, is folded, coiled, and looped into a nucleus that's only a few micrometers wide. And this three-dimensional shape isn't random. It's highly organized, extremely compact, and absolutely crucial for gene function.
Particularly for transcription, right?
Particularly transcription. If the folding process is disrupted, the instructions might not be readable, or worse, they might be read at the wrong time or in the wrong place.
Okay, that makes sense.
Precisely. The folding ensures that distant regulatory elements can physically interact. Two sections of DNA that are separated by millions of base pairs in the linear sequence can be brought into intimate contact inside the nucleus.
Which allows a regulatory element to activate its target gene or, I guess, repress it.
Right. And this organization exists at multiple nested scales, sort of like Russian dolls of structure.
Let's break down those scales using our origami analogy. What's the smallest, most essential fold, the equivalent of a crucial paperclip holding 2 pages together?
That would be the chromatin loop. These are the fundamental structural components. They serve two main purposes: compacting the genome and mediating these essential regulatory interactions.
And they do that by bringing distant elements close together.
Yes, like distal enhancers, which act as volume knobs for genes. The loop brings that enhancer right next to its target gene promoter. This physical proximity is what coordinates gene expression.
So without those loops, the volume knob is physically too far away to turn on the gene. That makes a lot of sense. So zooming out, what do a collection of these loops form?
You get the topologically associating domains, or TADs.
TADs.
Think of a TAD as a stable, insulated chapter of the genome. They are larger structures formed primarily by a mechanism called cohesin-mediated loop extrusion. The key functional point is that DNA sequences inside a TAD interact with each other much more frequently than they do with sequences outside that TAD.
That insulation seems critical. It will prevent an enhancer in one chapter from accidentally activating or repressing a gene in the next chapter over.
It's functional compartmentalization. It protects genes from unwanted regulatory noise. And if you disrupt the boundaries of a TAD, which often happens in cancer, you can accidentally activate an oncogene that was previously insulated.
I see. And then the largest, highest level of organization?
That's the segregation of compartments A and B. This is the highest organizational scale, where the entire genome is broadly segregated based on its activity level.
So, on and off.
Basically, compartment A is the active, open, decondensed euchromatin, the sections ready for transcription. Compartment B is the repressed, compacted heterochromatin, the sections mostly silent and tightly round.
And they stick together. Active with active, silent with silent.
These interactions, active regions binding with active and silent with silent, create that characteristic two-section checkerboard pattern that you see in contact maps.
It sounds incredibly intricate. How do scientists actually measure these fleeting physical contacts inside a tiny nucleus? I mean, it seems almost impossible.
It requires a pretty revolutionary chemical trick. The foundation is a technology called chromosome conformation capture, or 3C. And its high-throughput derivative, high-C, is what really provided the evidence for TADs and compartments.
Okay, let's pause and simplify that part again. What's the core principle of high-C?
High-C uses formaldehyde to chemically cross-link any pieces of DNA that are physically touching inside the nucleus at that moment in time.
So like taking a snapshot.
It's exactly like taking a snapshot. You then fragment the DNA, and importantly, you re-ligate the touching pieces chemically. So if piece X was touching piece Y, they are now physically linked into one piece.
Ah, I see.
We then sequence these resulting chimera fragments. The sequencing data provides a measure of contact frequency. The more often we sequence a link between X and Y, the closer they must have been in 3D space.
So the frequency of contacts in the sequencing data becomes the proxy for physical proximity, and that tells you the 3D structure. It's a method of converting spatial distance into sequencing counts.
You got it. And this concept has evolved into a powerhouse clinical tool called genomic proximity mapping, GPM, which is a specific high-C-based NGS assay. And this is where the 3D folding moves from foundational research into immediate, high-impact clinical diagnostics.
The power of GPM must be immense, especially in finding things that linear sequencing just completely overlooks. I'm thinking specifically of structural variants, SVs.
This is the key clinical application. Traditional linear sequencing is fantastic at reading the text. Single nucleotide variants, but structural variants,
Hmm.
large-scale deletions, duplications, inversions, rearrangements, they are fundamentally structural errors.
Right. If the folding of the paper is wrong, just reading the text linearly might not tell you where the creases are or where the paper has been ripped and pasted back incorrectly.
Exactly. So GPM, by detecting these 3D contacts, can simultaneously detect both copy number changes, so how many copies of a DNA section exist, and rearrangements where sections have moved.
The clinical results for this were stunning.
They were. In a study of 123 individuals with suspected genetic disorders, GPM achieved 100% detection of all known variants previously identified.
100%.
That's 110 deletions and duplications, 27 rearrangements, with perfect concordance to the established diagnosis. That really confirms its robustness.
But that's just validation. For me, here's where it gets really interesting. The novel discoveries.
That's right. Beyond just validating known conditions, GPM uncovered 12 novel structural variants that standard clinical tests had completely missed. These were complex, often cryptic rearrangements that conventional assays just weren't sensitive enough to find.
Can you elaborate on that complexity? You mentioned a case where a known translocation was found to be far more complicated.
Mhm.
Can you paint a picture of the difference for us?
Of course. So imagine a genetic test identifying a standard three-way translocation. Visually, that might look like three simple cuts and three simple pastes involving three different chromosomes. It's a clean diagram. But when GPM mapped the 3D proximity, that same patient was revealed to actually have 13 separate breakpoints across 4 chromosomes.
13 breakpoints. Wow.
The simple translocation was a superficial diagnosis masking this deeply intricate, cryptic structural chaos that fundamentally changes how the genome is regulated. And GPM found these additional cryptic changes in every patient examined who had multiple rearrangements.
That's a dramatic increase in resolution for clinical insight. If the genome is folded like paper, GPM is identifying all the tiny creases and tears that linear reading completely overlooks, giving us a true structural map of the genetic damage.
And because it relies on capturing proximity, it's also highly sensitive. It can detect low-level mosaic variants where a disease-causing rearrangement only exists in a small percentage of the patient's cells with very high accuracy.
That practicality is huge. I mean, GPM works on low-quality, archived, or preserved tissues and requires substantially less input DNA.
It does. It accelerates the move from simply finding a genetic association with the disease to providing genetic validation of the causal mechanism.
And if we accept that most complex diseases, like MS or IBD, are driven by risk variants in non-coding regions, these regions that affect when and where a gene is expressed, then the 3D organization is the essential missing link.
It is. GPM provides that integrated structural framework needed to define causal biology by physically linking the distant regulatory region to its actual target gene.
So we've established the crucial importance of 3D organization inside the nucleus. Now, WBSO needs to take that blueprint and map it across whole intact organs. This transitions us to the actual hardware and chemistry of capturing that spatial information across macroscopic tissues.
And this is where the technological trade-offs become really critical. When we evaluate any emerging spatial omics platform, we judge them by 3 standard metrics.
Okay.
Resolution, so the finest detail, gene complexity, the number of different molecules you can detect simultaneously, and gene sensitivity, the accurate detection range, making sure you capture low-expression genes.
But as we established for whole-body spatial omics, where we're looking at these massive tissues, we have to add a 4th defining metric: spatial depth.
Spatial depth. It's the extent of z-axis coverage. How deep into an intact, large sample can the technology reliably penetrate? And that depth requirement is the fundamental challenge that separates true WBSO from traditional 2D spatial transcriptomics, which, often just rely on thin slices.
Let's categorize the available ST technologies then, because they fall into 2 pretty distinct camps, each optimizing for different trade-offs.
They do. You have sequencing-based and imaging-based.
Let's start with the sequencing-based ST.
Please.
These are the unbiased approaches, right? They're designed to capture the entire transcriptome, everything expressed in the tissue,
Yeah.
and they rely on spatially indexed surfaces.
That's the one. The analogy here is that sequencing gives us the whole book, it's unbiased, but the image of the location is a bit fuzzier, a lower resolution. The most prominent example is probably the 10x Genomics Visium platform.
How does Visium work?
Visium uses these barcoded spots on a slide, which captures RNA from all the cells directly above that spot. The resolution is typically around 55 micrometers per spot, which means each spot captures the collective expression of about 10 to 20 cells.
So it's superb for whole tissue context and unbiased discovery, but it kind of smears the single-cell information.
It does, but they are rapidly converging toward single-cell resolution. Visium HD is their high-definition version, which moves resolution down to approximately 2 micrometers per pixel.
That's a massive improvement.
It's huge. It's approaching true single-cell resolution, but it generates proportionally massive amounts of data. Another high-resolution player is StereoSeq, which uses DNA nanoball arrays to get very high-resolution 0.5 to 2 micrometers, and it's known for capturing very large tissue areas. They've demonstrated imaging of an entire mouse embryo.
Wow. Okay, so sequencing-based ST is great for unbiased discovery and getting a complete molecular profile, but sacrifices some fine detail unless you move to these HD versions, which generate huge files. Now, let's look at the other camp, the imaging-based ST, or in situ methods.
Imaging-based methods choose precision over breadth. They are targeted. So they use fluorescent probes and highly complex sequential imaging cycles to detect specific, pre-selected gene panels.
Typically hundreds to thousands of genes.
Yes, but they offer dramatically better, often subcellular resolution.
So they give us sharp photos, high resolution, but only of the chapters we pre-select.
Exactly. Key examples here would be 10x genomic xenium, which has subcellular resolution and can detect panels of up to 5,000 genes. And then there's VisGen MERSCOPE, which uses multiplexed error-robust FISH. MERSCOPE achieves molecular-level precision with localization accuracy down to 100 to 200 nanometers and can assay up to 10,000 genes by cycling through multiple rounds of imaging and probe removal. Omics is another similar platform with high resolution for targeted panels.
These imaging methods sound superior for understanding cellular interactions because they preserve the cell's physical integrity and show where the molecules are within the cell itself.
Absolutely. Preserving cellular integrity and getting that subcellular resolution is vital for things like neurobiology and oncology, where the precise location of a signaling molecule within a neuron or at a tumor boundary dictates its entire function.
Now let's talk multi-omics, the essential integration. WBSO isn't just about RNA. We need protein and morphology too. How do these platforms reliably combine modalities?
They approach it through physical co-localization. For a platform like Visium, you can perform H&E staining, the classical histological stain or immunofluorescence, IF, for protein analysis on the exact same section of tissue used for RNA sequencing.
Okay, that makes sense.
The newer Visium CytAssist kit specifically helps transfer the tissue geometry onto the barcoded slide, which enables a really reliable integration of both RNA and a pre-validated antibody panel for protein from a single tissue section.
And Xenium also supports this multimodal approach, right?
Xenium integrates multi-omics beautifully. It can perform H&E and IF staining after the RNA detection run is complete. And crucially, they use a sophisticated multimodal cell segmentation mix.
So it's not just DAPI for the nucleus.
No, it includes antibody staining for the cell membrane and the cell interior. This multimodal approach provides highly accurate landmarks for automated computational cell segmentation, ensuring that the transcriptomic mapping is assigned to the correct cell boundaries.
This all sounds incredibly dependent on the quality of the biological sample itself. Let's address the practical challenge here. FFPE tissues are common archival samples, but their preparation often causes artifacts.
That's the cold, hard reality of working with clinical material. FFPE, formalin, fixed paraffin embedded tissues are the backbone of pathology. They're stable, easily archived, abundant. But the formaldehyde fixation creates chemical crosslinks that can degrade RNA and obscure protein epitopes, and the subsequent dehydration and rehydration steps. They can also cause morphological distortions.
So even though a technology like Visium or Xenium is compatible with FFTE, the user has to confirm the RNA is still of high quality before starting the run.
Precisely. You have to ensure the RNA quality index, the RQI, is sufficient. Furthermore, optimizing large antibody panels across diverse FFPE tissue samples, especially older patient samples, is a huge challenge. It demands substantial user expertise in pathology to ensure that any observed molecular difference is genuinely biological, not an artifact of epitope degradation.
The technology is advanced, but the foundational biology remains the most critical variable.
Always.
This highlights that even as we build these high-tech machines, the fundamental histology expertise remains essential. We've established great tools for 2D sections and near single-cell resolution. But if we want whole-body spatial omics, we still haven't solved that spatial depth problem. We have to see deep inside an intact large organ, and light simply won't let us do it.
Exactly, which means we need to get away from light.
Okay, here's where we move completely past the microscope as we know it. We're abandoning photons. We need methods that encode spatial information without relying on light-penetrating dense tissue.
This is the non-optical frontier, and the most revolutionary development here is the emergence of volumetric DNA microscopy, or VDM.
DDN.
VDM is a fundamentally distinct approach. It relies on converting the limits of light penetration, the optical problem, into a manageable, high-throughput computational data processing problem.
So it's a chemical encoding system, turning the entire sample into a measurement device. Let's walk through the complex mechanism slowly using a teach-back approach, because this is conceptually divorced from traditional imaging. VDM relies entirely on thermodynamic entropy and molecular proximity.
Okay, so start with the preparation. You chemically tag individual target molecules, RNA or DNA in situ, inside the specimen with unique synthetic randomized nucleotide sequences. We call these UMIs, or unique molecular identifiers.
So every target molecule gets a unique barcode and retains its original 3D location. What's step 2?
Step 2 is proximity recording using diffusion. These tagged molecules are amplified and then allowed to diffuse in place, forming these transient diffusion clouds around their original spatial point. When diffusion clouds from two different UMIs overlap, they are designed to undergo a specific concatenation reaction, which chemically links the two distant UMIs.
And this linkage is the measurement. So the closer two original molecules were, the more frequently their diffusion clouds will overlap and concatenate, right?
Precisely. This linkage is recorded with a new randomized sequence called a unique event identifier, UEI. The frequency of UEI formation, measured statistically, encodes spatial proximity. This is the heart of the method.
That is truly mind-blowing. The sample itself is chemically generating the data that tells us the distance between molecules.
Exactly. Step 3 is the computational challenge. We sequence the resulting concatenamers to read the UEIs, which yields a massive, sparse matrix of proximity data. UMI A linked to UMI B, UMI C linked to UMI D, and so on with a frequency count.
And the algorithm takes this matrix and has to solve a statistical inverse problem.
That's the global coordinate problem we touched on earlier. You're asking the algorithm to infer accurate 3D coordinates based only on this sparse list of who is chemically close to whom.
And for that, you need some serious math.
Yes. We use nonlinear methods, specifically geodesic spectral embeddings, GSE, to infer the relative 3D coordinates of the original UMIs. The algorithm finds the set of 3D coordinates that maximizes the probability of observing the sequence data we collected. It turns a chemical process into a geographical map.
What are the primary advantages of VDM for WBSO, considering it completely avoids light?
Well, VDM is inherently scalable and fully compatible with whole-mount imaging, which is critical for true whole-body approaches. Since the image is captured chemically and volumetrically, it entirely bypasses the limitations of light scattering and diffraction.
And it's equipment independent.
Relatively, yes. It relies on standard wet lab protocols and industrialized next-generation sequencing pipelines, which lowers the operational cost per unit volume compared to maintaining specialized high-power microscopes.
And the real-world application here demonstrates the potential. They mapped an entire living organism.
VDM was successfully used to create a complete 3D DNA image of a whole zebrafish embryo. And this wasn't a slice, it was the whole specimen. It provided an unprecedented view of genetic sequences and their precise location, down to the cellular level within the context of the entire developing organism.
It also showed multi-omics potential.
It did. It can be adapted to capture DNA, RNA, and proteins via oligo-antibody conjugates.
That's the chemical encoding path. Now let's look at a physical preparation technique that complements this by solving the resolution challenge in a simple mechanical way. Expansion omics.
Expansion omics, EXO, which is derived from expansion microscopy, is just ingenious in its simplicity. It uses swellable hydrogels to literally physically enlarge the biological specimen.
It just makes it bigger.
It just makes it bigger, expanding it isotropically, so uniformly up to 4 times linearly. That 4-fold linear increase means a 60-nanometer physical resolution is effectively achieved.
So instead of using specialized equipment to optically shrink the light's wavelength, you just make the sample 4 times bigger so that a conventional, cheaper microscope can see the fine details.
Exactly. This physical magnification overcomes the optical diffraction limit using conventional microscopes, making high-resolution imaging dramatically more accessible.
That brings up a great point about synergy. What's fascinating here is that VDM relies on chemical diffusion and GSE to measure distance, rather than photons. It's like turning the sample into the measurement device. How quickly can this VDM technology scale to, say, human organs?
Well, the scalability is theoretically very high because the measurement relies on the robustness of the NGS pipeline, which is already highly optimized. The current bottleneck for large, dense human organs is really twofold.
Okay.
First, reliably and uniformly preparing the tissue to allow the UMI copies to diffuse predictably. And second, the computational inference step, which grows exponentially in complexity with sample volume.
That makes sense. We need robust hybrid protocols, like performing VDM on an exo-expanded tissue, to physically magnify the effective resolution while maintaining chemical stability, and then we need the computational power to handle the output.
That synergy is the key to WBSO's future. If VDM's molecular encoding were performed on an expanded tissue, the effective resolution achieved by the proximity statistics would be physically magnified, offering a clear path to highly resolved and scalable whole-body mapping.
The moment we transition from imaging a small, thin slice to a whole zebrafish embryo, or imagine a whole mouse organ, the data volume just explodes. Our sources indicate that a whole mouse body light sheet fluorescence microscopy LSFM scan can result in 3.5 terabytes of TIFF images.
One scan.
That's one scan. And that volume is staggering. It's not just the size, it's the complexity. We're dealing with multidimensional data combining spatial coordinates, XYZ, sequencing information, A, T, C, G, and often multiple omics layers.
This creates massive computational bottlenecks that AI and specialized algorithms are urgently needed to solve.
The sources outline 5 critical computational challenges that have to be addressed to transform these terabytes of raw data into useful biological insight. Let's dedicate some time to fleshing out these 5 pillars.
Okay. First is the foundational issue, big data management. This means simply acquiring, storing, visualizing, and accessing these massive files reliably.
You can't even open the file.
Right. So this requires high-performance computing infrastructure, tools like Omero, and reliance on arrays of high-performance GPUs.
If you can't store it or look at it efficiently, the data is useless. What's the second challenge, which relates to cleaning the data?
That is data quality control. When you combine data from hundreds of sequencing runs or different slides and protocols, technical noise and sequencing bias are, unavoidable. Eliminating batch effects, so systematic, non-biological variation, is essential to ensure that differences observed are due to biology, not technical variability.
Third and fourth challenges revolve around translating the raw data into actual biological structures.
Third is annotation. This means defining the cell boundary, identifying specific cell clusters, neurons, immune cells, etc., and quantifying RNA expression accurately at single cell and tissue levels. And the fourth is interpretation. This is the higher level analysis, finding specific tissue-wide dynamics like cell-cell interactions in 3D space or RNA velocity.
And the fifth, which is arguably the most difficult problem we face in WBSO, is spatial multi-omics integration.
This is the current hotspot because it's the bottleneck to true causal discovery. We need methods to reliably combine disparate molecular datasets, genomics, transcriptomics, proteomics, especially when they are collected sequentially on separate, non-perfectly overlapping serial tissue sections.
This is the spatial diagonal integration problem.
That's the one.
Let's focus on the most difficult specific bottlenecks, starting with image analysis. Segmentation. It sounds foundational, defining the cell boundaries in 3D. Why is this so hard?
Because the simple assumptions just break down. Segmentation relies on clear membrane markers, but traditional methods often fail in crowded or expanded tissues, especially exosamples where cell membranes can become discontinuous due to the physical swelling process.
And if you can't accurately define a cell's boundary, you misassign its transcripts, corrupting the entire analysis.
Correct. So how do researchers define cell boundaries if the physical markers are unreliable? They are turning to computational inference. Solutions include graph-based frameworks like the Bearing tool, which leverage transcript co-localization graphs to infer where a cell must be located.
Based on the clustering pattern of its own RNA molecules.
Exactly. And even more radically, there are segmentation-free approaches, like Ficture, which bypass explicit boundary detection entirely and just model the spatial distributions directly. This is highly scalable for handling massive submicron resolution data.
That segmentation-free approach sounds absolutely necessary for VDM, given that VDM relies entirely on inferring 3D coordinates from sparse local proximity data. That inference step seems like the greatest source of potential error.
You've identified the global coordinate problem in VDM. The computational task is immense. Convert a massive sparse matrix of local proximity data who is chemically touching whom into accurate global 3D coordinates that map the entire specimen. Simple linear methods.
Like the initial spectral maximum likelihood estimation, SMLE.
Right. SMLE works locally, but it breaks down over large length scales because the geometry of a biological specimen is highly nonlinear.
Wait, so why does the simpler SMLE break down, and how does geodesic spectral embeddings, or GSE, specifically fix that long-range coordinate error?
That's a great question. Let's break it down. SMLE treats distance as purely Euclidean, so straight line. But in a folded, convoluted biological sample, the meaningful distance is often geodesic, the distance along the curved or convoluted structure of the tissue.
So if two cells are physically far apart but connected by a long, winding neural process, the Euclidean distance is large, but the functional geodesic distance should be small.
Exactly. GSE solves this by preconditioning the UEI matrix. It uses nearest neighbor analysis and eigenvector decomposition to robustly capture longer-range positional relationships and estimate these geodesic distances. This nonlinear approach significantly enhances accuracy over the massive length scales required for whole-body mapping.
So it's about acknowledging the curvature and internal structure of the biological space rather than treating it like a rigid box. That's clever.
It is essential, especially when integrating data from different slices, which brings us back to that spatial diagonal integration problem.
Right. If we collect RNA data on slice A and protein data on slice B, we have non-overlapping information. We need to align the molecular data from A onto the molecular data from B using some common reference point.
And that common reference point is often histology. AI-driven approaches are mandatory here. Frameworks like Spatial Glue, which uses graph neural networks, are designed specifically for multimodal integration. Crucially, they leverage the classic histology stain, H&E.
As a universal anchor.
Exactly.
That's fascinating. The oldest, most basic pathological tool, the H&E stain, becomes the key to integrating 3.5 terabytes of modern omics data.
Yes. The H&E stain captures the morphology, which is consistent across serial slices if the tissue processing is accurate. By successfully aligning the morphological features, the cell shapes, the tissue boundaries of all slices, Spatial Glue can then computationally align the molecular layers linked to those boundaries. It bridges the gap.
So if we successfully integrate all these layers of information, the 3D genome, the RNA, the protein, the morphology, we're not just seeing the cells, we're essentially defining their functional neighborhoods. Why is that neighborhood concept so key in disease progression and therapeutic design?
Well, the neighborhood defines function. Cells, especially immune cells, neurons, and tumor cells, they act in concert. They communicate constantly via chemokines, secreted factors, direct cell-cell contact. If we define a functional neighborhood, say a specific organized region where cancer cells are highly aggressive, or a specific suppressed immune niche deep inside the tumor parenchyma, we can understand precisely why a particular drug is failing or how a disease is spreading its influence.
The spatial context is the ultimate arbiter of cellular function.
And that function dictates treatment efficacy.
This incredible technical capability building these high-resolution 3D maps of folding and function has to translate into massive real-world impact. What's the immediate payoff for therapeutic discovery and ultimately for the patient?
The impact is transformational because it shifts drug discovery from focusing on statistical correlation to focusing on causal biology. As we discussed, for complex immune-mediated diseases like IBD or MS, most risk variants lie in non-coding regions that influence when and where a gene is expressed.
Traditional statistical genomics gives you association. 3D multiomics provides the integrated structural framework needed to define the causal pathway.
That's right. Conventionally, if we found a disease-associated genetic variant, we used to assume it affected the nearest gene linearly, but our sources show that assumption is wrong about half the time because of the folding.
Half the time.
Absolutely. The Turner team emphasizes that without the 3D context, conventional approaches waste immense resources prioritizing incorrect targets. 3D multiomics solves this by integrating genome folding data like GPM with chromatin accessibility and gene expression data. It maps the long-range physical interactions between regulatory regions and their actual target genes.
And this physical proof is what converts a genetic association into genetic validation. It dramatically accelerates the selection of high-confidence targets.
And this validation process requires a high-quality reference point. They're not starting by mapping the disease, they're building an atlas of health first.
Which makes a ton of sense.
Precisely. Research groups are building comprehensive multi-omic atlases of healthy cells, including their full 3D genome structure, across all major cell types and tissues. This establishes a baseline, what the regulatory landscape should look like in its normal functional state.
Then, disease-associated variants found by large-scale GWAS studies are overlaid onto this healthy 3D structure.
Correct. And that overlay immediately reveals where the normal gene regulatory relationships are physically disrupted by the disease-associated variant. It points directly to the causal pathways and the specific target gene that needs to be modulated. This genetically grounded approach eliminates the immense cost and time of pursuing incorrect hypotheses.
It must reduce failure rates significantly. Let's turn to oncology, where spatial context is paramount. Tumor growth and metastasis are fundamentally 3D spatial processes. How does WBSO change our understanding of the tumor microenvironment, or TME?
WBSO allows us to build a comprehensive 3D blueprint of the TME, moving beyond static 2D snapshots. We can analyze how tumor cells, stromal cells, and immune infiltrates organize and interact in 3 dimensions. This detailed 3D map reveals hidden complexities, like the architecture of blood vessels within the tumor or the spatial arrangement of immune suppressive cells.
I recall a powerful, specific application concerning drug distribution kinetics. This is the difference between a drug being chemically perfect and physically useless.
This is a perfect example of the power of the where. Advanced 3D imaging techniques have provided quantitative maps of macromolecular drug distribution within the TME, and what they revealed was critical. Anti-cancer antibody drugs, such as those targeting HER2 or EGFR, are often physically restricted.
How so?
They bind efficiently to their antigens near the tumor edge, essentially forming a binding sink or a barrier that prevents them from penetrating deeper into the tumor parenchyma.
So the drug uses itself up binding to targets on the outside layer, and it never reaches the cancer cells in the core. That insight would be functionally impossible to obtain from a flat 2D section.
Exactly. The spatial map reveals the drug's journey and its failure point. This insight into drug distribution kinetics is vital because it immediately suggests new delivery strategies, like smaller molecule versions or combination therapies designed to break down that peripheral binding sink.
Beyond cancer, WBSO is crucial for neurobiology, which is perhaps the most spatially complex system of all.
The brain is extraordinarily intricate, composed of over 5,000 distinct cell types organized into complex circuits. Subcellular resolution platforms like Xenium and MERScope are vital for studying complex neurodegenerative conditions like Parkinson's and dementia.
By mapping gene expression within the intricate architecture of the brain,
We can unlock the secrets of how protein aggregates form, how communication breaks down, and how specific spatially confined cell types degenerate.
so if we can summarize the strategic value, 3D multiomics accelerates development by focusing on high-confidence targets with causal evidence, dramatically reducing the immense cost and time of pursuing incorrect hypotheses in early discovery.
It is the next necessary step. Just as next-generation sequencing revolutionized genetics, 3D multiomics is revolutionizing functional biology. By integrating all molecular layers within a structural framework, WBSO illuminates the underlying mechanics of complex diseases that have resisted traditional approaches. It offers a credible path to the next wave of truly effective therapies.
This has been an incredible journey through the physics, chemistry, and computing that is allowing us to build the 3D blueprint of life. Let's quickly recap the 3 foundational pillars of 3D discovery we covered today using the concepts we introduced.
Okay. First, we established the importance of genome structure, driven by technologies like Hi-C and GPM. This is about understanding the molecular origami, the 3D folding of TADs and compartments, to pinpoint causal genes and uncover those cryptic structural variants that linear sequencing misses.
And that provides immediate clinical utility, turning genetic association into validation.
Right.
Second, we move to scalability. This is the non-optical frontier, using technologies like volumetric DNA microscopy.
VDM. VDM uses chemical encoding via thermodynamic entropy and molecular diffusion to achieve whole-body analysis, bypassing the light penetration limits that doom conventional microscopy in large tissues. We also highlighted expansion omics, XO, which physically enlarges the sample to achieve nanometer-scale resolution with standard equipment.
Now, the third pillar is data integration. This requires employing complex computational methods like the robust geodesic spectral embeddings, GSE, to accurately infer global coordinates from local proximity data in VDM.
And deploying AI frameworks like Spatial Glue using graph neural networks to reliably integrate multi-omics data across the third dimension, solving that spatial diagonal integration problem by using histology as a universal anchor.
Looking ahead, where is the immediate strategic focus for researchers in this field?
The immediate strategic focus is hybrid integration and standardization. We need standardized protocols that combine XO for maximum resolution with VDM for scalable molecular encoding. We also must rapidly accelerate the development of high-efficiency chemical anchoring strategies, specifically oligo-antibody conjugates, to reliably integrate proteins and lipids into this 3D nucleic acid map.
Moving WBSO beyond its current transcriptomic-centric view into truly comprehensive multi-omics.
Exactly.
This is all pointing toward a grand, unifying goal that sounds almost like science fiction.
The ultimate convergence of these standardized, highly resolved, and integrated 3D multi-omics datasets will form the essential foundation for building truly predictive biological models. Scientists are calling this the Artificial Intelligence Virtual Cell, AIVC. This is where we move beyond just mapping reality to simulating and predicting how a biological system will respond to external stimuli.
We've moved from simply reading the linear book of life to seeing how it's bound, how the chapters interact, and how those physical relationships change the story and the outcome of disease. As we continue to refine these 3D blueprints of the cellome, the next profound question for every cell we map is, if we can see exactly where you are, what you're doing, and predict what you'll do next based on your current surroundings, what happens when we precisely change your neighborhood?
Indeed. If we can map the city, the next step is city planning. It's a fantastic place to leave the discussion. Thank you for joining again.